Now in Chapter 15, we are going to discuss the digestive system, and here you can find the topics that we'll cover. First off, the function of the digestive system. You want to start the story from the top. So we'll start with eating the food, so ingestion of food and liquids, followed by digestion of these bigger molecules into smaller ones that our small intestine can absorb, and then absorption of all of these nutrients. And then the body will take the nutrients and start a whole metabolic transformation of them. So we could either store glucose, for example, as glycogen. We could use different things to make plasma proteins and so on. We can also detoxify things that came into the body. And all of that is really the function of the liver. So you kind of want to think of the liver as like the kitchen of the body, where it cooks up things that are needed by the body. And then things that we do not need, we'll get rid of. So elimination, the liver can do that as well by throwing things into bile. And then eventually we're going to defecate all of the waste products. And the digestive system, we usually don't think of it as having a function related to immunity, but it does have the acidity of the stomach, can kill some pathogens trying to invade the body through our food. We also have the appendix, which is lymphatic tissue, and there's scattered lymphatic nodules throughout the intestines. Now here's an overview of how digestion occurs. We eat macromolecules, which are big molecules, and then we start to break them down into intermediate products, which we still cannot absorb. It's not until we reach the end products of digestion, and these are the products that our small intestinal cells have the ability to absorb. So let's break it down one at a time. What about carbs? We eat either a polysaccharide or we eat a disaccharide. The polysaccharide would have to break down through with an enzyme known as amylase enzyme, and the very first amylase is found in saliva. So we start the breakdown or the digestion of carbs in the oral cavity using salivary amylase. Break down polysaccharide into maltose. So what happens to these disaccharides then? Well, we eat sucrose, which is table sugar, and lactose, and that is milk sugar. Again, these are disaccharides, so there are two molecules in there, and again, we have to break them down into their monomers. And that is done by these what are known as brush border enzymes, which are enzymes found within the small intestinal cells. And those are namely maltase, sucrase, and lactase that will break it down into their monomers or monosaccharides of glucose, fructose, and galactose. And these are the molecules that the small intestinal cells can absorb. In individuals that lack lactase enzyme, that will lead to the accumulation of lactose, which again as a disaccharide is too big to be absorbed. It is a sugar, so it will eventually ferment, leading to the formation of gas, diarrhea, bloating, and all of the signs and symptoms that are related to lactose intolerance. Now for proteins, those are polypeptides, and we want to digest them. These are the enzymes that do so pepsin, trypsin, and chymotrypsin. We get these peptide fragments, which are basically amino acids with a little two parts that we have to get rid of. We get rid of them through carboxypeptidases and aminopeptidases. We eventually end up with these amino acids, and those are the ones, again, monomers that the intestinal cells have the ability to absorb. As for the triglycerides, again, it's a glycerol molecule with 3 fatty acid chains. A lipase enzyme will break them down into a monoglyceride and 2 free fatty acids, and again, we have the ability to absorb those. Now, this is what kind of a breakdown of the intestinal tract wall, and you can see all of the different layers. This would be right here is the lumen of the small intestine. So this is the mucosa on the inside of the lumen. You can see the 4 layers. We have mucosa. Underneath it is a submucosa layer, and then a muscularis, and then a serosa layer. And the serosa layer is the visceral peritoneum, which is a connective tissue, which is the serous covering that lines the small intestine. And as you can see here, it will bring in the blood supply to it. So we'll start off with the mucosal lining. you've got the epithelial lining, and scattered within the epithelial, you'll find some glands. So we've got these mucus cells that obviously make mucus. You have exocrine cells, and you also have these what are known as enteroendocrine cells. So they are endocrine cells, meaning that they are going to be making hormones, but because they are in the intestine, we call them enteroendocrine cells. And remember when when we were discussing the endocrine system, we said that we have the main glands, but we also have cells that are scattered throughout the body, and these happen to be one of them. And then underneath the mucosa, you'll find the submucosa. You can find the blood vessels and the lymphatics in there. You can also find a plexus of nerves. So these lines right here are a nervous plexus called the submucosal plexus of nerves. And the submucosal plexus, if you look at where they end, they mostly end on the epithelium of the intestines. So these are going to increase or decrease the secretions from the mucus cells and the exocrine and endocrine cells. So a submucosal plexus stimulates or inhibits the secretions from these glands. Underneath that, you have your muscularis externa, which is the smooth muscle fibers. And when those contract and relax, it'll give that peristaltic wavy movement of the loops of the intestine. It's made out of an inner circular and outer longitudinal fibers. And you can also see another plexus of nerves known as the myenteric plexus. The myenteric plexus, myo means muscle, and that plexus is really responsible for controlling the contractions and relaxations of these smooth muscles. We already talked about the serosa. So a couple of things here to take away points that are important is that the submucosal plexus, when stimulated, will lead to the increase in secretions coming out of these glands, while the myenteric plexus, if stimulated, will lead to the contraction and the peristaltic waves that we see in our loops of intestine. Now, how are the GI processes regulated? Okay, so it's regulated by the volume and composition of the luminal contents, meaning how much did we eat and what did we eat? Okay, so the volume and composition of the contents of the lumens, whether it's the gastric contents or the intestinal contents. So rather than, it has nothing to do with the nutritional state of the body, which means that our digestive system is designed to digest and absorb anything that gets into it, whether our body needs it or not. Okay, so again, your digestive system is not going to think, well, you know what, this person does not need any more, let's say, any more cholesterol. It doesn't need any more protein or fat. If we eat it, the digestive system is going to digest it and absorb it. Okay. So the gastrointestinal reflexes are initiated by a couple of things that happen within the lumen. Really, the digestive system, actually, we tend to think of it as having its own nervous system, and now the textbooks are talking about it in the sense of an enteric nervous system. So remember, in the nervous system, we said we have central and peripheral, where now we're kind of thinking about it, well, there is like a third kind of nervous system related just to the digestive system, and that is known as the enteric nervous system. And it's regulated by a couple of things. For example, the distension of the wall. Okay, the more we eat, that will lead to the stimulation of stretch receptors, and that will lead to to increase in the secretions and increase in the motility of the walls. The chyme osmolarity, so the solute concentration of what we're eating. Is it too salty? Is there too much sugar? The chyme acidity, whether of what we eat or the gastric acidity. And then the concentration of chyme of specific digestion products. So is this meal mainly a protein meal, or is it mainly starch, and so on? All of these are going to change what kind of enzymes, digestive enzymes we need, and so on. Okay, if you're not familiar with what chyme is actually after we eat food and it starts to mix up with all of the fluids in the digestive system. It is now called chyme. So just like anything we have that is regulated, it is either under nervous control or under hormonal control. So we'll talk first about the neural regulation of the GI tract. We already said that we talked about the enteric nervous system, and we introduced the two nerve plexuses, the myenteric and the submucosal. We said that the myenteric controls the smooth muscles of the small intestine, while the submucosal plexus controls the secretions from the gastric or from the digestive system. Okay. In addition to that, there are also impulses that come from the autonomic nervous system, so from the higher entities, the CNS, and those are your either sympathetic or parasympathetic. Sympathetic is going to inhibit the digestive process, while parasympathetic will stimulate the digestive process. Remember, parasympathetic was your resting and digesting system. Now, both of these, sympathetic and parasympathetic, actually synapse or connect and control. They can control the myenteric and submucosal plexuses too. So we have what are known as a short reflex and a long reflex. So let's take a look at what on earth those means. So your short reflex means that locally within the wall of the intestine, for example, and I'm going to use another image in order to explain this as well. So let's explain it first and then go to that other image. So in the gastrointestinal wall, we have different kinds of receptors. So we have chemoreceptors that will pick up, the different chemistry of what we're eating. Is it protein? Is it carbs and so on? It can also, we have osmoreceptors, and that will pick up the osmolarity of our food. And mechanoreceptors that will pick up the stretch that is happening to the lumen. So how much food did we eat? Now, all of these are going to stimulate the local enteric nerve plexuses. So the myenteric that will stimulate the smooth muscles, and the submucosal plexus that will stimulate secretions from their glands. Okay, and that is obviously a response from your lumen. That is your short reflex. So again, I want to go back to that image. So this is where the chyme would be within the lumen. And again, we have receptors throughout the in the mucosal lining. They're chemoreceptors, osmoreceptors, and stretch receptors that are picking up different information about that chyme. And because of these local nerve plexuses, namely your submucosal, will increase the secretions from these glands, and the myenteric plexus will increase the contractions that are happening here in order to have that, again, peristaltic wave to move the chyme throughout the digestive system. So that is your short reflex, which is basically local within the GI wall, the gastrointestinal wall. Now for your long reflex, well, that would have to come all the way from the central nervous system. So for example, if we get hungry, or if you pass by a pizza shop and you can smell or see or even taste food, that is going to stimulate things within the hypothalamus, and then the hypothalamus is going to send impulses through the vagus nerve, which is your 10th cranial nerve. These are going to be parasympathetic impulses to your, again, local nerve plexuses within the GI tract. So again, your myenteric or submucosal plexuses, leading to the increase whether of the motility or increase of secretions locally. So your short reflex is really within the GI tract. Your long reflex comes all the way up from the central nervous system. Now for the hormonal regulation, The hormones that are secreted from the gastrointestinal system are secreted by these enteroendocrine cells, and those cells are scattered within the stomach and the small intestine. The 4 GI hormones that we really understand and we're going to be discussing here are gastrin, CCK or cholecystokinin, secretin, and GIP or glucose-dependent insulinotropic peptide. So a couple of things that we can learn just from looking at these names. Okay, gastrin is all about the stomach. Cholecystokinin, kinin means coming, you can relate it to kinetic energy, so it's going to move something, and it moves the gallbladder. So cholecyst, cyst is a bag form, and cholecyst means the gallbladder. So cholecystokinin or CCK is going to activate the gallbladder. Secretin, and we'll get to that when we move on to the next slide. Glucose-dependent insulinotropic peptide means that its presence is really dependent on the presence or absence of glucose. So let's take a look at more information on these. First off, for gastrin, things that are similar, they are all peptide hormones. Okay, for gastrin, as we mentioned, this is really related to the gastric area, so it's all about the stomach when we talk about gastrin. It is produced by the antrum of the stomach. If you're not familiar with what the antrum is, that is the lowermost part of the stomach. It's right before the pyloric sphincter. And it is the secretion of gastrin is stimulated by eating of protein and obviously the parasympathetic nervous system. What it does is that basically you're telling the stomach that food is now entering into the stomach. Okay, this person now started to eat, especially if it's a protein diet, and it will stimulate the secretion of acid, the hydrochloric acid, and it stimulates the motility of the stomach in order to help in digestion of protein. So protein digestion needs a lot of hydrochloric acid. It will also stimulate the motility of the small intestine and the large intestine. So basically, we're kind of emptying the small intestine and large intestine in order to prepare it to welcome, so to speak, the food, the new food that is being digested in the stomach. Now, gastrin is responsible for a reflex known as the gastrocolic reflex, which is a very interesting reflex. We get a lot of patients that would tell you, I really cannot empty my bowel unless I have something to eat. And that is because of gastrin. When they eat something, gastrin comes out from the stomach and it increases their mass movements of the large intestine, leading to a bowel movement. Again, a reflex known as the gastrocolic reflex. Now, what about CCK? Well, CCK is secreted by the small intestine, and its secretion is stimulated by the presence of amino acids and fat in the small intestine, which means we are now beyond the stomach. Okay, so food is now being emptied from the stomach. It's going into the duodenum, which means we can stop, the stomach can stop making acid and can decrease its motility. So CCK is going to inhibit acid secretion and stomach motility. But what it does do is that it stimulates insulin secretion and stimulates the contraction of gallbladder and relaxes the sphincter of the gallbladder that is known as the sphincter of Oddi. So again, you have to kind of imagine the scenario, which is right in this image. This, you can see here, the chyme, full of acid and full of fat or amino acids, and being emptied into the duodenum, which is the first part of the small intestine. That is going to stimulate the cells here to make CCK. CCK will circulate in the bloodstream, as you can see here, and reach the gallbladder. CCK will contract the gallbladder that is full of bile. Now bile is going to be released, and CCK would also lead to the relaxation of that sphincter known as the hepatopancreatic sphincter or the sphincter of Oddi, and then bile will enter into the duodenum and help in the digestion of that chyme that comes in here. Again, we're emptying the stomach, so another thing CCK does would be to inhibit the gastric secretions of hydrochloric acid and motility. Now, secretin is all about the acidity. Okay, it is another peptide hormone that released by the small intestine. Its release is stimulated by the acidity, whether it's the hydrochloric acid or whatever we're eating, mainly the hydrochloric acid. And it will inhibit the gastric acidity, it inhibits the gastric motility. And because secretin is stimulated by that acidity coming from the stomach, it stimulates the pancreatic secretion of bicarbonate in order to neutralize the gastric acidity. Now, GIP is another peptide hormone made by the small intestine. It is stimulated by the presence of glucose in the small intestine. So basically we're eating something full of sugar, and its function is to stimulate the pancreas to make insulin. So again, one of the things that is nice with its name is that it tells you what it does. Gastric glucose-dependent insulinotropic means that it releases insulin hormone. Okay, so I apologize for going back and forth, but there was no way to have this in one page. So now, what are the phases of gastrointestinal control? When we eat, there are different phases to the eating process. So there's a cephalic phase, which is basically everything that is going on in your head. So am I hungry? I can smell food. I can see food. That is known as your cephalic phase. So it's all in the head. That would be in order to stimulate the digestive system, the autonomic nervous system through parasympathetic neurons, again, that are carried, or parasympathetic stimuli that are carried on the vagus nerve, are going to increase the secretions and increase motility. So that again is your long reflex. Now that we're actually eating, we will move on to a gastric phase of control. And that is when the stomach starts to distend. And again, it depends on what we're eating. So are we eating mainly proteins or is it mainly starch? And that is going to lead to the release of the gastrin hormone due to which is mediated by your short and long neural reflexes. So those local reflexes within the gastric wall will lead to the release of gastrin secreted by the stomach. And then when the food or chyme moves on to the intestines, that's when we move on to the intestinal phase, and that would be mediated by your short and long neural reflexes through these hormones that we talked about, CCK, secretin, and GIP. And though all three of those hormones, if you remember, were released by the enteroendocrine cells of the small intestine. Okay, so this ends this portion.